Hello everyone, and welcome to today's webinar, HbA1c Past, Present, and Future. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Professor Philippe Gillery, Professor of Biochemistry and Molecular Biology at the Faculty of Medicine of Reims, France. Professor Gillery, you may now begin your presentation.
So, welcome to this webinar devoted to HbA1c. During this lecture, we will address three topics. First, we will have a look on the past and complex history of HbA1c, and then we will review the current challenges of HbA1c, analytical and clinical ones. First, the complex history of HbA1c and reference of papers that you can read for additional information. HbA1c is one of the most powerful biomarkers discovered and implemented in clinical practice during the last 5 decades. The heterogeneity of human hemoglobin was demonstrated during the late 50s, which allowed to separate minor hemoglobins called fast hemoglobins due to their behavior, or HbA1, made of different fractions, HbA1a to HbA1e. Their origin was puzzling for many years. And in 1968, an abnormal hemoglobin was described in red cells of diabetic patients, and this so-called abnormal hemoglobin was HbA1c. Then the structure of HbA1 and especially HbA1c was identified. It was demonstrated that HbA1 fractions were formed from HbA by a reaction called non-enzymatic glycation. They are also called amidary products since they are formed during early steps of this reaction, which corresponds to the spontaneous binding of sugars and especially glucose and their byproducts on amino groups of proteins. Among HbA1 fractions, HbA1c, as shown in this example of old chromatogram, is the major fraction. It was shown that HbA1c was formed by binding of glucose to N-terminal valine residues of globin beta chains, but please note that glycation may occur on other sites. It is formed by a cumulative and irreversible process during all red blood cell lifespan of about 120 days, and it depends on glucose concentration. Here is schematically represented the structure of HbA1c. On the left part of the slide, you can see a molecule of HbA with 2 alpha chains and 2 beta chains, and the list of the preferential glycation sites. The most glycated site is the beta and terminal valene of globin beta chains, and this corresponds to HbA1c. But other sites can be glycated on both chains, alpha and beta. And another factor of heterogeneity is the possibility that glucose binds to one or two N-terminal beta chains, but also on one or two N-terminal alpha chains. So the term HbA1c covers a number of molecular forms, and this underlines the necessity of a strict definition of HbA1c for the standardization of assays. Quickly after HbA1c discovery, HbA1c has become the gold standard of diabetic survey. It was demonstrated by two large-scale epidemiological studies, the DCCT study for type 1 diabetes and the UKPDS study for type 2 diabetes. Both studies showed the relationship between HbA1c and degenerative complications of diabetes mellitus. It was proved that HbA1c was a reliable marker for the optimal control of glycemic balance, HbA1c being a retrospective and cumulative index of glycemic balance during the 4 to 8 weeks before sample. At that time, reference values, decision limits, and therapeutic targets were established. But HbA1c values used in clinical practice were established first using many methods based on different principles and with variable analytical performances, and second, with reference to the NGSP DCCT standardization program, which was an international program mainly based in the United States, and using a non-specific reference method for HbA1c measurement. And indeed, before standardization, the situation of HbA1c assays was relatively chaotic. There were many disorders in terminologies and concepts, and the performance of methods were highly unequal. You can see on the right part of the slide the results of the first quality control assessment that we performed in France in 1995. And for that purpose, we sent one sample to more than 2,000 laboratories for measuring HbA1c. The target values was 6%, but you can see that the results from laboratories ranged from 5 to 11%. In addition, the imprecision of methods was very high, being very rarely below 5% and very often at 10 or even 20%. Thus, there was at that time an indisputable need for standardization of HbA1c assays. This standardization was internationally managed and achieved by the International Federation of Clinical Chemistry and Laboratory Medicine, or IFCC. In this standardization process, the measurand was clearly defined as the beta and terminal hexapeptide. So the glycated hexapeptide was related to the non-glycated, and HbA1c was expressed in millimoles HbA1c per mole HbA0, which is the major peak of HbA, plus HbA1c. The reference measurement procedure was also described. Hemoglobin was digested by a specific enzyme, which allowed to quantify specific hexapeptides by LCMS coupled to mass spectrometry or capillary electrophoresis. This reference measurement procedure, or RMP, is maintained by an international IFCC network of approved laboratories, and this network constitutes a valid anchor, especially for calibration of field methods by manufacturers. The missions of this network are to guarantee the continuity of the IFCC RMP and to make HbA1c assays worldwide traceable to the RMP. Now, we can see that after 15 years of experience, there is a good stability of values. And now, all routine methods are traceable to the IFCC RMP worldwide. This HbA1c standardization is a worldwide process with unique anchor. But the description of this method had a side effect, which is a change in HbA1c value reporting. Indeed, the IFCC RMP specifically measures HbA1c, contrary to previously used national so-called reference system like NGSP in the USA using ion exchange chromatography. HbA1c values obtained by the IFCC RMP when expressed in percentage of total hemoglobin are about 20% lower than those obtained by the NGSP method. So there was a risk of confusion, and in order to avoid it, IFCC proposed to change value expression millimole HbA1c per mole HbA0 plus HbA1c instead of percentage of total hemoglobin, which is an expression in SI units. It was possible to establish a correlation between the two types of expressions using a master equation. And of course, a new challenge was the implementation of new HbA1c numbers. For example, using classical units in percentage, the reference range considered at that time from 4 to 6% of total hemoglobin corresponded to 20 to 42 millimoles HbA1c per mole hemoglobin. In this respect, it was important to have a strategy of implementation of these new numbers, and this was made by an IFCC committee, but there is still now a reporting of units different according to the countries. Sometimes dual reporting, sometimes only SI reporting, or only NGSP reporting. So there's still a need of education and of communication with clinical societies and patients. However, what could be observed during all this period is a continuous improvement of method performances, as described in many papers. Some examples are here. And you can see on the slide, these are the results of the CAP survey in the United States, that with time, the results obtained by the different methods were more and more close to the target value, and that the imprecision of the method greatly decreased. Thus, the international standardization of methods by IFCC and the improvement of methods by manufacturers made a robust support for new applications of HbA1c assays. Now, we will focus on the current challenges of HbA1c assays, and first, analytical ones. Just a word on the methods used today for measuring HbA1c. They can measure HbA1c or total glycated hemoglobin, but in both cases, due to the traceability to the IFCC RMP, all results may be expressed as HbA1c. Methods measuring HbA1c can use chemico-physical properties. This is the case of cation exchange chromatography by HPLC or capillary electrophoresis, antigenicity for immunoassays, or structural properties for enzymatic assays. And affinity method, performed either by HPLC or automated assays, measure total glycated hemoglobin. Well, we can say that now there are less and less relevant concerns due to technical improvements of the methods. We can now use well-controlled, automated and standardized methods. Identical HbA1c values are obtained due to the standardization. With most methods, there are few interferences. Some of them can be used in POCT and in clinical settings, and there is a global improvement of performances together with the development of large-scale quality control trials. Anyway, there are still two types of analytical challenges. First one, how to measure and to evaluate method performances, and then how to establish quality targets. If we go back to this slide showing the results obtained before standardization, you remember that we had very different values and that the conclusion was the need for standardization. But the situation observed was not only a matter of method performances. Why? Because a more recent concept has appeared, which is the importance of commutability of control materials in external quality assessment schemes. When we considered these results in the first approach, the conclusion was, of course, that methods exhibited different analytical performances and that some of them were acceptable and others not acceptable. But if we have a more in-depth approach, we can suspect that some differences were also due to the non-commutability of the materials used. Commutability of control materials is a mandatory need for reliable information. A simple definition of commutability could be the property of a calibration or control material to behave like a fresh patient sample with a given technique. And the importance of using this type of materials is underlined by recommendations, for example, from the ISO 15189 for external quality assessment schemes. In this equals, an important point is to establish quality targets. There are different ways to establish quality targets, but one of the most attractive one, in my opinion, is that proposed by IFCC and the group led by Dr. Cas Feikel. This expression is related to the relation between the trueness, which is the bias of a given method when compared to a reference method, in function of the imprecision expressed by the between laboratory CV. And this representation allows to easily see which methods are close to the best goal, which is close to zero bias and zero CV, hardly reachable, and it's possible to evaluate the performance of methods in terms of bias and of imprecision. The same group has organized a European trial for HbA1c measurement, and they sent samples to more to 2,000 laboratories in 17 countries. The materials sent were issued from single donations and sent to the laboratories under three conditions: fresh, frozen, and lyophilized. In parallel, these samples were assayed by reference laboratories, which allowed to establish targets for the measurement. This study demonstrated that there were variable performances for HbA1c assays according to countries on the top of the slide, according to manufacturers below, but also on the type of samples. And this was more precisely described by an experiment that we made some years ago, aiming at determine the trueness of HbA1c routine assays, and in order to know if processed EQA materials were up to the job. So we assessed commutability of 26 processed quality control materials using 17 of the most frequently used HbA1c assays. These materials were sent to manufacturers, expert laboratories, and also to medical laboratories. This slide shows some results of this study. On the top, it confirmed that methods had different performances in terms of bias and imprecision, and this scheme represents the results obtained with fresh, whole blood. And in the lower part of the slide, we could show that there were differences between fresh and processed materials. Here you have the results on the left, you have the results of fresh clinical specimens, in the middle of frozen clinical specimens, and on the right side of the panel, processed EQA materials. So you can see the difference of results according to the type of sample used. And then on the right panel, you can see that various results were obtained using different methods, and this shows the non-commutability of some materials. It shows that if the materials are not commutable, it is not possible to evaluate method performances, but it just demonstrates that the material is not convenient for quality control trial. Thus, the consequences of non-commutability are the inability of EQA schemes to evaluate the ability of the different assays to provide equivalent results. And in all cases, commutability of EQA materials should not be assumed. It should be evaluated systematically when the goal is to evaluate bias of assays and or agreement between results provided by the different assays. If commutability has not been evaluated, the best option is to use materials consisting of fresh whole blood, of course, if possible, because this is much more constraining. Now, the clinical challenge of HbA1c. There are two types of clinical challenges now for HbA1c. First, the situations where HbA1c results are not interpretable. This is the case, for example, when there are modifications of red blood cell lifespan and alterations of hemoglobin metabolism, anemias, transfusions, hemolysis, troubles of iron metabolism, or some therapies. There are also situations where there are discrepancies between markers of glycemic balance or competition between reactions, and in order to illustrate it, we will take two examples: hemoglobin variance and renal insufficiency. The second type of challenge is related to the new clinical use of HbA1c. Classically, HbA1c was used for the survey of glycemic balance, but now HbA1c can be used for diagnosis or screening. So first, the situation with complicated interpretation. Again. First, interference of hemoglobin variance. On a pathophysiological point of view, Hb variance may cause hemolysis and thus alter lifespan, which also alters the semiological value of HbA1c, and all methods, of course, are concerned by this point. For the analytical aspect, it depends on the method, because Hb variance may interfere with the assay, and for other methods, Hb variance may pass unnoticed. When should we suspect the presence of an hemoglobin variant? This could be an analytical argument, but this depends, again, on method characteristics. Because non-separative methods, like automated methods, immunoassays, enzymatic methods, affinity, do not evidence the variant. And in the case of separative methods, ion exchange HPLC or capillary electrophoresis, the variant may be evidenced, or in some cases not. So there are very different situation according to the variant. There are also clinical arguments which can be evoked during the result interpretation. And a major argument is a discrepancy between HbA1c values and other results. HbA1c value can be lower than expected, but also can be higher than expected. Of course, other reasons of discrepancy must be eliminated, for example, errors in self-monitoring results. Here are examples of interferences in capillary electrophoresis. A normal electrophoregram is shown on the top left part of the slide with HbA1c, the major peak of HbA0, and minor peaks of HbA1 and HbA2. On the right part, you can see an example of HbC, which is easily separated from HbE0, and in this case, HbA1c value can be calculated. This is not the case of HbHope, which migrates very close to HbA0, and in the case shown, no HbA1c result can be calculated. But in some cases, the troubles may be much more subtle, and this is the case of delta variant shown here as a very minor peak, lower than 1%. In this case, HbA1c can be calculated. So there are various aspects and levels of interference, and in all cases, the question is it possible to issue a reliable HbA1c result? Here is shown a second example, represented by the interference of HbS in sickle cell disease with different methods. There is no interference with IFCC reference method by LCMS, and also you can see on the lower and left part that with affinity HPLC, the variant is not detected and there is no interference. This is not the case with capillary electrophoresis or HPLC cation exchange HPLC, where the peak is easily detected. So once again, there are various types of interferences depending on the method. And again, the question is it possible to issue a reliable HbA1c result? In order to address this concern, how to interpret and issue a result or a comment if it's not possible to have a result, for the most common variants, there are databases which can help us. For example, the database of NGSP, the National Glycohemoglobin Standardization Program, which gives very interesting information about the most common interferences. You can find here tables with the different methods. Here I have mentioned as an example only the principle of the method, but on the website are mentioned the names of the commercial techniques used, and you can see the interference from HbC, HbS, HbE, HbD, elevated HbF, and this can help you to interpret most of the cases. When there is a rare variant, information can be provided by literature and manufacturers, and there are so many variants that we cannot go into detail today in this lecture. However, in any case, if a reliable HbA1c value cannot be obtained, we have to use alternative markers like fructosamine or glycated albumin if available. Second example now, renal insufficiency. In patients with chronic kidney disease and renal insufficiency, HbA1c may be used, but result interpretation must be cautious because of several specific conditions of these diseases. First, there are modifications of red blood cell lifespan due to chronic anemia, but also to possible erythropoietin treatment. Another phenomenon is the binding of other adducts to hemoglobin. And this is a case of carbamylation of hemoglobin. For many years, carbamylated hemoglobin was an analytical concern because it interfered with many methods. But this is no more the case, and now most methods have improved to eliminate this interference. But this is still a pathophysiological concern. Just a word about carbamylation. Carbamylation corresponds to the enzymatic binding of isocyanic acid to proteins. Isocyanic acid is a natural byproduct of urea, which spontaneously dissociates in cyanate and isocyanic acid. This isocyanic acid is very reactive and binds to proteins. And this process is increased in patients with chronic kidney disease because the increase of urea. So the question is it an interference of carbamylation on hemoglobin glycation? In fact, there is a potential competition for the same hemoglobin sites. Both isocyanic acid and glucose are able to bind to the beta and terminal valine, giving either carbamylated hemoglobin or HbA1c. So does carbamylated hemoglobin formation interfere with HbA1c formation, and which are the consequences on HbA1c values and result interpretation? For addressing this question, we have performed an experiment in mice. We used diabetic and uremic mice and measured in different conditions HbA1c in order to determine if there was a competition between glycation and carbamylation. Here are the results. We can just look at the left panel. HbA1c was measured in control mice and CKD mice. HbA1c values were identical, about 5%. In diabetic mice, HbA1c was much higher. All these results were expected. But what is especially interesting is that in mice with diabetes and renal insufficiency, HbA1c values is much lower than in mice with only diabetes. This demonstrates that carbamylation significantly inhibits glycation in vivo. And here we have plotted hemoglobin ANC values against carbamylated hemoglobin values in two models of mice. And we demonstrated that when carboxylated hemoglobin increased, HbA1c decreased. Thus, carbamylation competes with glycation for HbA1c formation, and thus the interpretation of HbA1c results in patients with chronic kidney disease must be very careful. Now, to finish, the new indications of HbA1c, this is a change of paradigm because for years and decades, HbA1c was only used for monitoring diabetes and as a biomarker of diabetes follow-up. But now, HbA1c is also recommended for screening or for diagnosis, and there are recommendations from WHO, the American Diabetes Association, and several national guidelines. And with HbA1c, it's possible to evaluate the level of risk from a low risk, when HbA1c is lower than 39 millimole per mole or 5.8%, to diabetes when it is over 47 millimole per mole or 6.4%. There are advantages for assaying HbA1c instead of glucose in this case. First, and this is important, it is not needed to obtain fasting samples. Then there is a lower variability of HbA1c than of blood glucose. HbA1c represents an integrated reflector of glycemia, and there are internationally standardized methods with a reference network. However, there are some inconveniences. The results of HbA1c may be subjected to interferences. We have seen the examples of HP variants and red blood cell metabolism alterations. The cost of HbA1c values is higher than that of glucose, and variations were demonstrated according to the ethnicity. This has to be confirmed, but it could lead to the establishment of different thresholds depending on the population. However, now HbA1c, with its role on diabetes follow-up and diagnosis, can be considered a global biomarker of diabetes. Now, 5 take-home messages. HbA1c is the major glycated hemoglobin and is formed by a non-enzymatic reaction depending on glycemia and red blood cell lifespan. HbA1c assays are standardized worldwide. They are traceable to a reference measurement procedure maintained by a network of reference laboratories. HbA1c assay methods exhibit generally good performances, but EQA schemes must prove the commutability of control samples used. HbA1c is used not only as a long-term retrospective marker of glycemic balance, but also as a diagnostic marker. And HbA1c results are interpretable in most cases, but must be cautiously considered in specific clinical situations, presence of an HP variant or renal impairment. And this leads us back to the first sentence of this presentation. HbA1c is one of the most powerful biomarkers discovered and implemented in clinical practice during the last 5 decades. Thank you for your attention.
Well, thank you, Professor Gillery, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Okay, let's get started. Find my questions. So our first question, is it possible to discover a hemoglobin variant in a patient followed for many years by HbA1c without any biological sign?
Yes, this is a frequent case which highlights the necessity to know precisely the characteristic of the technique used. As I said, some techniques are able to detect variants, others are not due to their principle. In such a situation, the characteristics of the techniques used must be carefully checked. And in most cases, the explanation is that HbA1c was initially assayed with a non-separative technique and then with a separative technique able to detect the variant. Thus, the choice of the technique is a specific prerogative of every laboratory specialist who must be aware of the advantages and limitations of the method used. This does not presuppose about the intrinsic analytical qualities of the method, but it determines the correct interpretation of results.
Well, thank you, Professor Gillery. Our next question, are there genetic factors influencing HbA1c values?
This is a puzzling point, which is not fully understood. Well, I repeat, this is in fact a puzzling point, which is not fully understood. It seems that some patients have a higher glycation rate than other ones. Thus, it's classical in literature to oppose slow or low glycators to fast or high glycators. Thus, a future strategy could be to screen specific diabetes-associated genes in order to adapt target values of HbA1c, but these are still undergoing investigations, and this is not yet in current practice, but this is an interesting perspective.
All right, thank you again, Professor Gillery. Our next question. How is it possible to survey diabetic control in patients homozygous for one variant? For example, a patient with sickle cell anemia may have diabetes.
Well, in this case, obviously, HbA1c cannot be assayed because there is no HbA. Besides, hemolysis may occur according to the type of variant and modify red blood cell lifespan, and the result is that the semiological value of HbA1c is greatly decreased. So in this case, the best solution is to assay other biomarkers of glycemic control, mainly fructosamine or glycated albumin. These markers provide information on a shorter period, 2 to 3 weeks before the sampling, but they allow a correct follow-up in one given patient.
Looks like we have time for one more question. What are the perspectives of HbA1c use in diabetes survey in the future?
Well, this is a very important question. I think that HbA1c will remain the gold standard of glycemic balance, especially in patients with type 2 diabetes, where glycemic variations are relatively limited. In the case of type 1 diabetes, it is a little bit different because there are more glycemic excursions, glycemic variations. Thus, other strategies may be used for assessing glycemic control. One of them is the use of continuous glucose monitoring. These systems allow a more dynamic view of glycemic changes, and they are more and more frequently used by diabetologists in patients with type 1 diabetes. However, these systems are not yet standardized, and their standardization remains to be achieved.
Well, thank you, Professor. Do you have any final comments for our audience today?
No, I hope that the messages were clear and that the audience was happy with the content of the presentation and my choices for illustrating the topic. And I thank you.
Well, thank you again, Professor Gillery, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Abbott, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.